Today's class is different in the sense that you have to listen to it on your own time, and in class we will have a quiz to determine whether you have done your homework or not, and we will discuss whatever you didn't understand and what your thoughts are on this topic by doing some activities. Today's topic is epigenetics, which is actually my favorite topic in the field of genetics. The reason for that is that it shows how your environment, your stressors, your eating habits, and even the ones of your parents and grandparents can impact you at a very molecular level without actually changing the genetic information. Basically, it represents some sort of a record of how your genome and your ancestor's genome have interacted with their experiences and their environment. There are a few objectives that I'm hoping to achieve by the end of the lecture. First and foremost, you need to understand the concept of epigenetics and how it modulates gene expression. There are various mechanisms that operate at different levels, and unless I'm saying otherwise, you need to understand how these epigenetics modifications can actually be achieved. Also, you need to understand the concepts of genetic imprinting and X chromosome inactivation, and how both nutrition and stress can have an impact on gene expression. Findings on how epigenetics can actually impact oral health are still in their infancy. However, I hope to give you some food for thought and enough knowledge to determine the validity of any new findings that will come out in the future. So what is actually epigenetics? The term was coined for the very first time in 1940s by Conrad Waddington, and in direct translation it means above genetics. Epi in Greek means above. What we actually mean when we talk about epigenetics are actually all the changes in gene expressions that can modify or determine a phenotype, sometimes passed on to future generations without being inscripted in the DNA or without making any modifications in the DNA sequence. The formal definition, established in 2008, was stably heritable phenotypes resulting from changes in the chromosome without alterations in the DNA sequence. Those epigenetic changes can either be stable and transmittable to the offsprings, or they can be dynamic and temporary, constantly added and removed from the genome. Epigenetic adds another layer of complexity to gene regulation, which is already difficult as it is, and trying to understand and determine the epigenetic status of an individual is actually quite challenging. The reason for that is because each cell in each organ can have different epigenetic markers. Also, all these epigenetic markers are very susceptible to different environmental factors. They can be very transient. So unless you actually catch them at a specific time, you cannot determine the exact epigenetic status of a person. And also, the epigenetic status can change drastically with age. All these epigenetic markers there are a lot of epigenetic markers in the early stages. They sort of stabilize towards the middle age, and towards old age they start to be removed. So why study epigenetics? It is a fascinating topic to me, and I hope for you too, and it actually settles the old age debate of nature versus nurture, of whether we are defined by our genes or by our environment, and it veers it towards nature via nurture, in the sense that both our genes and our environment have to contribute to determine who we are. The best models for how a phenotype can be changed without any changes in DNA are homozygotic twins which are genetically identical. And the perfect example you can see in this chromosomal stain of a set of the same twins at the age of 3 and 50. Epigenetic markers for one twin are labeled in red, and for the other twin are labeled in green. At the age of 3, there is a lot of yellow, you can see here, meaning that there is a lot of overlap in their epigenetic markers. However, at 50, you can see that they differ quite drastically depending on their own individual experiences. You can also see that two identical twins can end up looking quite differently, and they can have different pathologies, especially when it comes to diseases that have strong environmental components. Therefore, if you want to look at it as in an analogy, nature provides the music sheet. So the DNA is basically the music sheet. And however, in order to interpret the music and to produce music, which is basically the phenotype, you need a conductor to give its own interpretation, and you need musicians, which are the genes or the mRNA, to produce the music. So this is, I think, or at least in my opinion, it is a very good analogy about how all these levels work together in order to produce a finite product. So this is a schematic concept of epigenetics. Basically, environmental factors, which involve diet, smoking, our inflammation status, our age, can change the epigenetic status by various modifications, which we will talk about, such as acetylation, methylation, in order to modulate gene expression and to influence various phenotypes and pathologies. So let's see what the different epigenetic modifications are. These markers can be added either at the level of DNA or at the level of chromatin. At the level of DNA, the only known modification is cytosine methylation. At the level of chromatin, however, there are a few modifications that can occur: methylation, acetylation, phosphorylation, and ubiquitination of histones. Also, recently, non-coding RNA have been considered part of the epigenetic toolkit. Non-coding RNA are transcribed, however, they are not translated, and they can operate at various levels, either by blocking translation or by binding to mRNA to target its degradation, which is the case of microRNAs. Long non-coding RNAs are actually starting to become a very hot topic in the field lately. They are longer RNAs. They can reach up to hundreds of thousands of nucleotides, and they have been shown to regulate gene expression, and the most famous example for that, for long non-coding RNA regulation, is X chromosome inactivation. Another way to think about all these epigenetic markers is as if they are traffic signs on the road, and they coordinate the traffic to stop, slow down, accelerate depending on the needs of the environment. First, let's take a look at DNA methylation. This is probably the most common epigenetic modification. It is basically the addition of a methyl group to the C5 of a cytosine, and with formation of 5' methylcytosine. Methylation is catalyzed by DNA methyltransferases, DNMTs for short, and it is the methyl group is again provided by SAM. The targeted cytosine has to be a specific cytosine, specifically one that is neighboring a guanine on the same DNA strand. So since the cytosine and the guanine are neighboring each other, they are bound by a phosphodiester bond, and therefore these sites are known as CpG dinucleotides. CG dinucleotides are the ones that pair by hydrogen bonds from different DNA strands. So please be careful and not mistake CpG dinucleotides with CG nucleotides. DNA structures that are rich in these dinucleotides are called CpG islands. These sites are usually located on or around the promoters and result in silencing and stabilizing these DNA regions. So, for example, if a gene is active without the methyl group, methylation on the cytosine on its promoter can repress its activity. DNA methylation is always acting as a repressor. However, there can be one instance in which it might look like it's actually activating gene expression. I want you to think about what that instance might be, and we will talk about it in class. Let's now take a look at DNA methyltransferases. There are two major classes, de novo and maintenance. De novo methyltransferases add methyl groups to previously unmethylated cytosines. They are part of the DNMT class 3. And that happens mostly during embryonic development or in response to environmental agents. Maintenance methyltransferases, DNMT1, maintain the methylation pattern on replicated DNA. Remember that DNA is replicated in a semi-conservative fashion with an old and a new strand for each new helix. The parental strand carries the original methylation pattern, and the daughter strand is initially unmethylated. So the DNA immediately after replication is hemimethylated. However, methyltransferases, DNMT1, will add, or maintenance methyltransferases, will add corresponding methyl groups in order to match the original methylation pattern, and in the end, the newly replicated DNA will become fully methylated. How do methyl groups act as gene repressors? Well, they can do that by two different mechanisms. One, it is very straightforward. Basically, the methyl group on the cytosine in the promoter region is physically preventing the binding of a transcription factor so that it can act as a transcriptional modulator. The second mechanism is a little bit more complex, and it involves the recruitment of certain proteins to the sites of methylation. These proteins are known as methyl CpG binding proteins, or MPBs for short. They need to, once they bind to the methyl groups, they recruit some other proteins, mostly histone modifying proteins, specifically histone deacetylases. And these histone deacetylases modify the neighboring histones and change the chromatin status, making it inaccessible for transcription. So these are the two ways in which methylation of cytosine on DNA, on DNA can actually repress gene expression. I mentioned at the beginning that some of these epigenetic markers are transitory and they can be easily removed. So how are these methyl groups removed when the transcriptional status of a gene needs to change? DNA demethylation, which is the process of removing methyl groups from DNA, can be achieved by two different methods. One is an active method where 5-methyl cytosine is converted to 5-hydroxymethyl cytosine by enzymes in the family of 1011 translocation proteins, which are TATs. These methyl cytosines can undergo further oxidation to a product, which is 5-carboxycytosine or 5-formyl cytosine. Either one of those can be recognized by base excision repair as a modified base and is excised and replaced by an unmethylated cytosine. Another option for active demethylation is to basically just deaminate 5-methylcytosine to thiamine, which will be excised and replaced by an unmethylated cytosine by mismatch repair. These mechanisms are both very controversial among scientists as they do not necessarily agree on all the specifics of the process. However, passive demethylation is generally agreed on. This means that demethylation occurs simply by lack of maintenance following replication. For example, if DNMT1 is inactivated, or if methylcytosine has already been oxidized to hydroxyl methylcytosine, and it's not recognized as methylated cytosine by DNMT1 during maintenance, and therefore no corresponding methyl group is added on the cytosine on the other strand. One example of DNA methylation is genomic imprinting. Let's see what that means. We know that humans are diploid organisms that have 2 alleles, 2 copies of one gene for each genetic locus. One inherited from the mother and another one inherited from the father. In most cases, both of these are expressed or can be expressed, and the phenotype is independent on which parent the allele comes from. However, there are a few genes for which the sex of the parents that contributed the allele can influence whether the gene is expressed or not. This process, in which the sex of the parent that transmits the allele can determine its expression, is called genomic imprinting. You can see here in this diagram that only the paternal allele is actually expressed. So, for example, if you have a mutated paternal allele, it will result in a mutant phenotype because that mutant gene is actually expressed. However, if the mutant allele is derived from the mother, where the maternal allele is permanently silenced, the offspring will be perfectly normal. There are a few genes that are imprinted in humans. However, they are quite rare, about 100 in the 22,000 in our genome, and mostly they're involved in growth and metabolism. For example, the insulin growth factor 2 is one of those imprinted genes, and it is expressed only on the maternal allele. However, the insulin growth factor 2 receptor is also an imprinted gene, and only the maternal allele is expressed. Also, there's an interesting example here, which you don't need to know for the exam, but it's actually quite funny. If you have, there's another perfect example of imprinting of breeding horses with donkeys. So if you have a male donkey and a female horse, they will produce a mule. If you breed a female donkey and a male horse, it will produce a hinny. And both of them, although they are absolutely identical at the DNA level, they have completely different phenotypes. So this is in general the concept of genomic imprinting. This process of genomic imprinting was discovered due to 2 different genetic disorders that have very different phenotypes. But it was discovered that they actually have exactly the same deletion on chromosome 15. In this region of chromosome 15, there are a few imprinted genes, and depending on whether the maternal or the paternal allele is mutated, you can get very different phenotypes. So, if the deletion actually occurs on the paternal allele, the result is Prader-Willi syndrome, characterized by mental retardation, obesity, hypotonia. However, if the maternal allele is the one that is deleted, the syndrome that the corresponding syndrome is Angelman syndrome, which is also characterized by mental retardation. However, it's also characterized by poor appetite, weight loss, and movement disorders. So although they have exactly the same genetic makeup, there are very different phenotypes, pathological phenotypes that can occur in response to that deletion. So now that we're done with epigenetic modifications that occur at DNA level, let's move on to the next type of epigenetic modifications, those on histones that impact chromatin compaction, and they are also called chromatin remodeling modifications. The most important one is histone acetylation, which is conducted by histone acetylases, HATs. It involves the addition of an acetyl group to histones, specifically to lysine and arginine residues, which confer usually a positive charge to the histones. These positive charges of histones are what helps them stick to the very negatively charged DNA, and once these positive charges are masked by acetyl groups, without those positive charges, the interaction between the histones and the DNA loosens, and the DNA becomes more accessible for transcription. Removal of these acetyl groups is done by histone deacetylases, HDACs, which we have shown before that they can combine with methyl binding proteins to amplify the repressive response of DNA methylation. So histone deacetylases remove those acetyl groups so that histones regain their positive charge, and they bind again very tightly to the DNA and end up closing the chromatin structure. The other two important histone modifications are methylation and phosphorylation. Methylation occurs by addition of a methyl group, this time not to DNA, but to an amino acid on histones. The effect of this methylation and the enzymes that are used for adding these methyl groups to the histones depends on whether the targeted DNA is an arginine or a lysine. So methylation on a lysine is conducted by histone lysine methyltransferases, and on arginine by protein arginine methyltransferases. Also, the result of methylation, depending whether it's on a lysine or an arginine, it can be either gene transcription activation or repression. Histone phosphorylation usually occurs on the usual amino acids, serine, threonine, and tyrosine. Once they are actually on once they phosphorylate those amino acids, chromatin is loosened and they promote gene activation. Phosphorylation, those phosphate groups are added by the regular histone kinases, or they can be reversed by histone phosphatases. Phosphorylation usually occurs in response to DNA damage. So, for example, if you have a double-strand break, the histones that are in the vicinity of the damage response genes are immediately phosphorylated, and they're activated in order for those genes to deal with the double-strand break and to repair the damage. There are also other modifications that can occur on histones, such as ubiquitination, sumoylation, crotonylation. However, all of these are very minor, and they're definitely not the target of our lecture. So we finally reached the third mechanism for epigenetic modifications, which is through non-coding RNA. We have here the most famous example of this type of modifications, which is the X chromosome inactivation by long non-coding RNA. This process occurs only in female females because they carry two X chromosomes, as opposed to males who have an X and a Y. The Y chromosome is extremely small, and it carries only a few genes that are all responsible for sex assignment. However, the X chromosome is very large, and it carries thousands of genes that can play very diverse functions in the body. Therefore, in order to compensate for females having double the genetic expression as males, in other words, in order to preserve the genetic dosage, which we will talk about in a different lecture, one of the X chromosomes is permanently turned off. The mechanism involves a long non-coding RNA, which is known as XIST, and this XIST is randomly expressed by one of the X chromosomes and immediately coats that chromosome. It is to no surprise that it will coat the chromosome that it expresses it, because long non-coding RNAs are very unstable and they get easily degraded without having the chance to travel further distances. So once XIST is bound to the chromosome, it recruits different proteins, specifically from the polycomb repressive complex, PRC, which again recruit histone methyltransferases, which will methylate lysines on histones and will result in full compression and shutting down of the chromosome. This inactive and highly condensed chromosome can be easily seen under the microscope in the cell nucleus, and it is called a bar body. The process of X chromosome inactivation is also called lyonization, after Mary Lyon, who has first described it, and it occurs randomly on either chromosome in each cell of the body. It can be easily exemplified by the random distribution of the orange and black pigment in the coat of the calico cats. So, for example, if you have an X chromosome, if the father passes an X chromosome with an allele for a black black pigment, and the mother passes on an X chromosome with an allele for orange pigment, due to the random inactivation of the X chromosome in each cell, the pattern of calico female cats is completely random, while the males are always black. So think now of whether, if you were to clone a calico female, would you get an absolutely identical clone to the original, or will they be different? Think about that, and we will talk about this in class. So now that we know about the different epigenetic modifications that can occur, let's see how they are passed on to the next generation. For the longest time, it was thought that epigenetic modifications were not heritable. However, in the last decade, it was discovered that DNA methylation can be passed on up to 3 generations. During fertilization, both the egg and the sperm carry multiple epigenetic tags, and most of them are removed in the zygote so that the embryo can actually start with a clean slate. However, there are a few instances in which DNA methyl groups are actually passed on to the offspring, and specifically those are the ones on imprinted genes, which are always passed on, and the other ones are the some of the methyl groups that escape this wipeout. We will talk about these examples in the next few slides when we discuss the impact of nutrition and stress on the phenotype of a person. So how does the epigenetic status change depending on the environment? Specifically, we will look at nutrition. A large study was conducted on children born in 1945 in the Netherlands during and after the Dutch hunger winter. This Dutch hunger winter was a period of about 3 to 4 months during World War II, when the Germans imposed a blockade on the Netherlands and cut all their food supplies. Also, it was a very harsh winter, and that combined with the lack of food resulted in a point where the maximum caloric intake per day for a person would be about 400 calories, and even those were not very nutritious. They usually came from grass or branches or tulip bulbs. However, despite all these shortcomings in food, the Dutch public system was very thorough and kept very accurate accounts on all the births that have occurred during that period. Also, the beauty of the study was that the period was very clearly defined for a few months, and also the government kept clear accounts on the food rations, on the official food rations, and so they knew exactly how much, when the women were pregnant, and how much their daily food intake was. So scientists that were involved in this study have noticed that children that were exposed, that were in utero during this period of low caloric intake, had long-lasting consequences during their entire adult life. However, it depends on whether they were in utero, on the age of the pregnancy at the time of the famine. So women that were exposed to the famine during their late trimester, during late gestation, so in the third trimester, they had babies that were significantly smaller than normal. And this small size was maintained even during the adult life of these babies. They were always shorter than their peers, and they're always shorter, always smaller. One good example for that is the actress Audrey Hepburn. If you Google her, you can see that she was always a petite woman, and she was one of these Dutch hunger winter children. However, there is the other situation in which the mothers were malnourished during the time of conception or during the first trimester of the pregnancy, and were well fed after. And they had, even though they were well fed during the last 2 trimesters of the pregnancy, their babies were normal sized. However, during their adult life, they usually become obese, and they would develop cardiovascular disease with a higher incidence than the normal population. The reason for that is, if you remember from our previous slides, was that during the first trimester is when the methylation pattern of the embryo is established after starting with a clean slate at conception. If you have low food resources, specifically of foods that were rich in folic acid, which is one of the major donors of methyl groups, you will end up with an embryo that is hypomethylated. So this study has shown that about 15 genes that are involved in metabolism, specifically in glucose metabolism, were hypomethylated throughout the life of a person that was deprived from nutrients during the first trimester in utero. This offers a very good explanation to the high incidence of metabolic syndrome in these people. It was actually shown that the methylation status can recover sometimes, and in mice, there is this gene called the agouti gene, which is usually methylated. However, if the mice were given a drug that would wipe out the methylation pattern, so the mice were unmethylated on the agouti gene, they were obese and prone to diabetes. Mice with methylated agouti gene are usually normal and healthy. So feeding unmethylated agouti mice a methyl-rich diet can recover their methylation status, and they will have healthy and normal weight pups. So this study shows how the nutrition status of a person can change its epigenetic status, and how it can actually have long-lasting impact on the health of a person for a long period of time. So now let's take a look at the impact of stress on the epigenome, and for that we use a licking and grooming rat model. It was about 5 years ago at a conference where I heard this scientist from Montreal talking about this subject, and I thought it was fascinating, and hopefully you guys will find it interesting too. So before we actually look at how stress impacts our gene expression, we need to first see how stress actually works. So whenever we are confronted with any kind of stressful situation, the stress circuitry is turned on and it activates the fight-or-flight response and induces the secretion of very high levels of cortisol in order to help with this stressful situation. However, too much cortisol is not good for health because high levels of cortisol can induce heart disease, can increase the risk for depression and cancer, and they can increase the inflammation in the body. Cortisol binds to glucocorticoid receptor, and when all the spots are filled, so enough cortisol is bound, the stress signals are turned off and they're shutting down cortisol production. Normally, glucocorticoid receptor is hypermethylated during embryonic development because it is thought to be evolutionarily advantageous to be a little bit more anxious and therefore more cautious in situations where there is high competition for resources. So usually during evolution, those individuals that were more anxious had higher chances of survival and passing on their genes. However, we are living in a situation where this competition for resources is absent. So the higher level for the higher chances for survival are for people that are dealing better with stress. And it was shown that rats and also people that have higher levels of glucocorticoid receptor are better at detecting cortisol, and they're recovering from stress better and with less permanent damage. Now that we established how the background of how cortisol and glucocorticoid receptor work, we should look at the experiment at hand. So if pups were born to a mother, to a rat mother that was very attentive, was licking and grooming them and nurturing them, the methylation on the glucocorticoid receptor was decreased, and the pups were calmer. You can think of it almost as if once the mother was licking and grooming them, it was almost like figuratively like she was licking the methyl groups away from the glucocorticoid receptor, allowing more of it to be produced and for the pups to deal better with stress. However, pups in a litter where the mother was neglecting them, was not licking and grooming them, were anxious and difficult to handle, and their levels of glucocorticoid receptor were very low. However, this experiment can be also replicated by taking a low nurtured rat, injecting its brain with a drug that removes the methyl groups, and it starts acting exactly like a high nurtured rat. The opposite works too. So if you take a high nurtured rat, a relaxed, calm rat, inject its brain with methionine, which is a methyl donor, it instantly becomes more anxious and stressed. Also, if a neglecting mother, has a litter of pups, and half of these pups remained with the neglecting mother, and the other half were given to a mother that is very nurturing and is licking and grooming them often, the rats that remained with the neglecting mother were anxious. However, the rats that were transferred into a caring environment, they became instantly calmer. So you can see here that stress has a very clear impact on gene expression at the very molecular level. It is difficult to transfer all this data and all these findings to humans because we don't want to abuse anybody. However, there was a very small study that was conducted a few years ago looking at brains donated by donated from suicide victims. And these brains came from individuals that were either abused early in their life or they came from very loving, nurturing homes. And it was shown that the methylation status of glucocorticoid receptor in those individuals that came from abused homes was way higher, so they had a very low expression of glucocorticoid receptors, unlike the suicide victims that came from a very loving, nurturing home. So although this study is very small, it has a very small sample size, it actually is showing very clearly that the findings in rats, in licking and grooming rats, are directly applicable to humans as well. And stress has a very strong impact on us at the genetic level. Next, we will look at the correlation between epigenetics and cancer. In the next lecture, we will talk more about oncogenesis. So all these things might make a little bit more sense then. But until then, you need to know that there are two different types of proteins that are contributing to uncontrolled cell proliferation. So for ones are the tumor suppressor genes, which are prohibiting that uncontrolled proliferation to actually happen. And then there are the proto-oncogenes that can be mutated and turned into oncogenes, which are stimulated uncontrolled cell growth, which is cancer. So when we look at epigenetics, if we have methylation on a tumor suppressor gene, that tumor suppressor gene is turned off and it doesn't do its job, and it allows basically the proliferation, the uncontrolled proliferation of mutated cells, and therefore cancer formation. The other situation that can occur is the demethylation of proto-oncogene, which allows for transcription factors to bind this promoter and allow the transcription of this proto-oncogene. And therefore, it triggers a series of events that will eventually lead to uncontrolled cell growth and cancer. There are two examples here where epigenetics actually can play a role in cancer formation. You don't need to remember them, but the two are in breast and ovarian cancer. Some breast and ovarian cancers result from the hypermethylation of the BRCA gene. The BRCA gene, if it's hypermethylated, it's turned off, and it's actually a gene that is involved in DNA damage repair. So once you don't have the gene, the DNA damage accumulates and it triggers uncontrolled cell growth. The other type of cancer that is correlated to epigenetics are some types of colorectal cancers that result from hypermethylation of MGMT gene, which is another gene that is involved in DNA damage repair. So basically the same mechanism applies in both cancers, where a gene that is involved in repair, in DNA damage repair, gets turned off and it doesn't do its job and allows mutated cells to proliferate. Now we finally reached the end of our lecture. We are looking at epigenetics in dentistry. However, not much is known so far. There are very few studies, and most of them, if you go to PubMed and search for epigenetics in dentistry, there are very few primary research studies. Most of them are actually reviews. And in reviews, very little details are offered on how the studies were conducted. It's more a philosophical approach on how epigenetics can actually impact oral health. But regardless, let's take a look at what is known so far, and I'm sure that in a few years things will actually develop more. So what we know is basically that the majority of problems that happen in oral in dentistry and in oral health are problems related to inflammation. Basically, the lack of balance between pro-inflammatory and anti-inflammatory agents. So we can see that there are epigenetic mechanisms at play in the development of the tooth structures, in development of different orofacial cancers, and also in the maintenance of this balance between inflammation and anti-inflammation, which results in periodontal disease and sometimes caries. So let's take a look at what the current findings are in the field of epigenetics that correlate to oral health. I don't want you to remember all these things. They won't be on the exam. However, it is good to be informed and good to know what the current findings in the field are. So first of all, we look at periodontitis. A study that looked at chronic periodontitis had pretty much the same findings as another one that looked at aggressive periodontitis. And in both cases, the balance between pro-inflammation and anti-inflammatory factors was tilted towards pro-inflammation. So therefore, they found that some interleukins were overexpressed due to hypomethylation on their promoter. And some other genes, such as tumor necrosis factor and COX-2, were inhibited because of hypermethylation on their promoter. Another field where where epigenetics is relevant is during dental pulp regeneration. There are two classes of histone deacetylases inhibitors that are currently tested. They're in clinical trials. And they have been injected to the sites of pulp removal, and they showed to be stimulating the synthesis of osteopontin and bone morphogenetic factor 2, stimulating the regeneration of pulp and mineralization process. Another area is in oncogenesis. For example, a study has shown that silencing of a tumor suppressor gene is actually the reason for one form of squamous cell carcinoma of the oral cavity. Another interesting area where epigenetics actually can correlate with dentistry is in enamel formation. Amylogenin, which is one of the major players in enamel formation, is a gene that resides on chromosome X. So if there is incomplete inactivation of one of the X chromosomes, the levels of amylogenin are higher than normal, and that translates into defects in enamel formation. Also, last but not least, we can think of how epigenetics can play a role in behavior management. For most people, going to the dentist can be a very stressful situation. So if we know that glucocorticoid receptor plays an important role in stress control, we can somehow think of a drug that a person can take before coming to the dentist, which will increase its level of, which will reduce its level of level of methylation on the glucocorticoid receptor, increase its expression, and therefore making for a more calm person and a more pleasant experience when confronted with the dentist. So this is the conclusion of our lecture for today. It was shorter than usual, and I tried to keep it to the bare minimum. However, we will have a lot more to talk during class, and I would like to hear your thoughts about how the environment that we live in can actually have an impact on our genetics.